Hello, everyone, and welcome to today's webinar hosted by Jen and sponsored by Roche. Today's topic is dPCR versus qPCR, selecting the right tool for molecular analysis. Thanks so much for being here. I'm Uduak Thomas, Senior Editor with Jen, and it's my pleasure to be your host for today's webinar. Before we get started, I'd like to give a little bit of background on today's topic. Real-time PCR or qPCR and digital PCR are both powerful molecular biology techniques for quantifying and analyzing DNA or RNA. However, they differ in their approach and applications. Now, traditional qPCR is widely used for detecting and quantifying specific DNA or RNA sequences in both research and clinical diagnostic applications. However, dPCR offers greater precision and sensitivity and is designed to handle tasks that require precise quantification. Today, I'll be speaking with Dr. Jim Huggett, Director of Biological Metrology and Visiting Professor at the National Measurement Laboratory, School of Biosciences and Medicine, and Boshen Gao, Support Scientist at Roche Sequencing and Life Science. We're going to discuss the dPCR workflow and the technology's advantages over traditional qPCR, including its higher precision, flexibility, sensitivity, and true absolute quantification. We'll also go over some of the differences between the technologies, including the resources and equipment that are needed to perform dPCR experiments and some of the pitfalls that you should look out for. Our discussion will also include types of applications that are best suited for the technology. Before we start the discussion, I would like to let our audience know that we'll be having a live Q&A session following this discussion. So if you have a question at any time, please enter it in the Ask a Question panel on your screen. All right, Jim, Boshen, thank you both for being here.
Good to be here.
Thank you.
Excellent. Let's start off with this first question. I think to set the ground for our discussion, we should start off with some of the fundamental principles behind digital PCR and a little bit about how those differ from qPCR. Jim, why don't we start with you?
Certainly. Okay. So I guess if we think of qPCR as a method that allows you to measure nucleic acids, you amplify nucleic acids using PCR reaction, but you include some kind of mechanism to measure the amplification, usually fluorescence, either using a double-stranded binding dye or a probe. Hydrolysis probes commonly used. And this is done in, we say, real time, in that the thermocycling is coupled to a fluorimeter, and you actually measure the increase in fluorescence as a surrogate for the increase in DNA that occurs in a qPCR. That is conducted in a liquid reaction, obviously, that can be 5 to 20 to 50 microliters, but it's all together in one reaction. And so the DNA is amplifying, it's increasing the concentration, and you're measuring that increase. Digital PCR is similar to qPCR, and it uses essentially the same reagents. And to be fair, I think it's fair to say that the developments of qPCR have allowed digital PCR to be able to, to be working as well as it does, because the reagents are fantastic. But it differs one crucial way, in that you're not conducting the PCR in a bulk reaction. You are partitioning it with a limiting dilution, so that the DNA molecules are kind of getting their own partition. And so you take a large volume, 20 microliters we're calling large in this context, and you partition that down into thousands, tens of thousands of very much smaller volume partitions. So you're having lots and lots of little reactions within these partitions, some of which importantly contain no DNA at all. And so while you can in some cases conduct real-time digital PCR, you don't have to. What you do is you conduct the PCR, and at the end of the PCR, you measure the partitions that have contained DNA as an increase in peak fluorescence against those who have nothing in them, which is the baseline fluorescence. And you count the negatives over the total number, and you can apply Poisson corrections to predict those partitions that contain more than one molecule. And so you're counting the partitions that contain DNA or increased PCR amplification over those who haven't. And so that's the fundamental difference with qPCR, whereas qPCR, you measure an amplification curve and you set a threshold across that amplification curve, which to predict the quantification cycle or cycle threshold, which is how you quantify using qPCR.
So that was really comprehensive. Boshen, I just wanted to come to you if you wanted to add anything to that description.
Yeah, I think Jim hit on the main principle of digital PCR, which is partitioning the reaction down to mini reactions, tens of thousands, sometimes hundreds of thousands. So this allows the reaction to be free of, well, to reduce the effect of inhibitors and also reduce competition for resources. And also the key is to capture the endpoint signal instead of in the process like qPCR. So this is what enables what we call true absolute quantification.
Thank you both. And Boshen, I'd like to stay with you for this next question. If we could still kind of talk a little bit about some of the differences here. What are the differences in terms of the instrumentation itself that's required for dPCR versus qPCR?
Yes. So because the digital PCR relies on partitioning, so you would need upfront an instrument that performs the partitioning process. And then with the reactions being subdivided into mini reactions into the partitioning, you can move that to a analyzer where it will perform PCR just like a regular qPCR instrument, and then it will be analyzed in the software.
And obviously, these are instruments that are being used in a laboratory. So Jim, I'm going to come back to you for this one. What are some of the implications, perhaps, if any, of these differences in the kinds of instruments that you're using for dPCR versus qPCR? How does that impact the way labs are set up and operated? If you could respond to that.
So I guess in the setup of the laboratories, we'd always recommend separating your pre and your post PCR, regardless of the format you're using. If I talk to and things are changing quite rapidly, that's a very exciting time for digital PCR because there are a number of solutions. But we look back over the last 15 years. One of the beauties of qPCR was it was automated. You set up your experiment, put it on, press go, and you wait for the results, and then you could analyze. Now that was a real advantage. We didn't have to run gels anymore. That means that qPCR is often used in diagnostics, for example, even when you don't want to quantify because of that automation. I guess when the newer form, digital PCR actually predates qPCR because you can do it with a 96 or 384 well or plate. But with the new and the renaissance, we call it, of the instrumentation, probably around 15, 16, 17 years ago, we took a step backwards because with qPCR, you set up your experiment in an 96 well plate and you put it on and forget about it. Well, for digital PCR, you need an instrument, or you need some format. I realize keep up myself to partition. You've got to take that reaction and you've got to do some partitioning, and that can be a chip-based fixed format or some kind of emulsion strategy to do the partitioning. And so that's an additional step. And so you need to consider where you're going to put that initial step. We certainly did know some of the newer instruments are getting rid of the need for that. So that was an additional complexity. And then again, I have to be careful here because the instruments are changing so quickly, but some of the instruments require a post after analysis step because you may need to read you will need to read. The question is whether that's automatic or requires an additional format step. So these are additional considerations, whereas in qPCR, it really is automated. There's little you need to do. And the beauty of qPCR is you have the option of analyzing, like doing post analysis, like melt curve analysis. There are some formats in digital that allow you to do that, but most do not. And so those are kind of setup differences to consider, or analytical differences to consider. But really, I think whether Boshen agrees with me, the actual setup of the instrument, really it's about making sure that your setup, where you set up the experiment, you handle the pre, is kept separate from the post. Now, because all PCR works by generating tens of billions of molecules of the very analyte you're interested in, And digital PCR especially is measuring single molecules of the analyte you're interested in. So it makes the perfect contaminant. So really, really that pre and post is absolutely essential for most setups. I mean, really.
Yeah, like keeping pre-PCR and post-PCR separate is, I would say, is one of the most crucial steps in setting up a molecular biology lab. And because the digital PCR instrument, the partitioning engine and the analyzer are separate instruments, so it's a little bit easier. I've seen setups where, you have the partitioning engine on one end of in a separate room and having the analyzer in a different room to keep it physically separate. And also keep in mind, because it's two instruments, so you can have multiple partitioning device set up to the same analyzer. This will increase your throughput.
Very interesting conversation, and we're definitely going to talk a little bit more about some of the innovations that will be coming out hopefully in the coming years in the future in this space. But still kind of sticking to some of the differences here, I wanted to ask, how do dPCR and qPCR quantitate nucleic acids differently? Boshen, why don't we start with you for this one?
Yeah, so I think that's one of the key differences, whereas qPCR captures the fluorescence signal continuously through the thermocycling program. The digital PCR is looking at the endpoint, so that enables absolute quantification without relying on things like standards, and this is why the name digital come from, where you're looking at just positive versus negative partition, and that's 1 versus 0, so that gives us the name digital.
And Jim, did you want to jump in on this question too?
But certainly, and the term digital actually works quite well in the reverse, because qPCR is actually analog. The qPCR amplification plot, maybe many of the audience can picture this. It is an amplification plot that looks a bit like a bacterial growth curve, for example. And you have to select where along that amplification plot you're going to draw a line. I mean, most analysts draw a line to set the threshold and calculate the quantification cycle or cycle threshold. And the cycle threshold, or CT value, is commonly used in COVID-19 as a measure, but it is highly variable. I mean, it's so highly variable that people almost cannot believe how variable it can be. And if you think of where the most advanced applications of molecular quantification with qPCR are, either in foods analysis, for example, for food adulteration, or in viral load testing, or measuring transcripts of BCR-ABL, for example, you really need to think about your calibration. This is a major consideration for qPCR, because the CT value or CQ value, it can vary within laboratories by hundreds of fold, by which I mean a given measure, 25, for example, a CQ value of 25 can vary in one lab. It could be, it could be 100 molecules. In another lab, it could easily be 10,000 molecules. So it is, how I put on it, it is highly variable. So when you switch to digital PCR, as we'll come to in a minute, you're counting these partitions. You really do have a much better measurement in terms of the need for calibration and whatever. So that is a major consideration, I think, when you compare the different analytical processes. qPCR can be, you can compare different amplification, the same amplification from different specimens. You should never really compare different amplification plots from using different assays. People do all the time, but they should not. And calibration, if you want to convert to copies, really is essential if you're going to use qPCR. It's much simpler with digital PCR, as I think we'll come to.
Just from my experience, and I believe everyone who has worked in a molecular lab, not just from a lab to lab difference, you can even see variation in the CT between same brand, same manufacturer's instrument, from instrument to instrument. So, but by employing digital PCR, you can definitely reduce this laborious process of validating, calibrating for instruments to instrument variability run lab to lab variability.
I'm going to come to we're going to talk about advantages a little bit later in terms of these technologies. But for now, what are some of the limitations of digital PCR as compared to qPCR? I think we may have started to touch a little bit on this question. Jim, I'll start with you.
No problem. I'm glad we've come to this actually, because I often get accused. I mean, I use both qPCR and digital PCR in our laboratory, and I'm quite passionate when it comes to PCR generally, and could easily be accused of being down on qPCR, and I'm not. I think qPCR is really important, ability to do very high throughput measurements. I mean, just imagine what would our response have been to the COVID-19 pandemic if we didn't have qPCR? I mean, it's slightly worrying to even think about that. So qPCR is a fantastic method. It has a huge dynamic range, which again, digital PCR can't really compete with. I mean, digital PCR's dynamic range with the instruments we have now up to maybe 100,000. I think Boshen correctly mentions there are some methods with more than 100,000 partitions, so they can get a bit higher. But you do not have the dynamic range you do with qPCR. And again, qPCR is well established. Lots of support for that. Everybody, lots of people know how to use it. A lot of people use it incorrectly. Just let that hanging in there. But an amazing method. And I guess my final point was, I believe it is qPCR and those manufacturers out there who really developed the reagents to allow qPCR to have really, really good linear dynamic ranges across vast, on log scale, vast dynamic range and very low limit of detections, that has made our switch to digital PCR possible. Because I remember starting, to my age here, qPCR 20 years ago, maybe, my PhD, and getting those reactions to working out you needed carrier, what polymerase is a good hot start. These are all headaches, and it was a mess. So the manufacturers really have got really delivered some fantastic reagents, which I think the digital PCR community has been able to embrace, and that's why digital PCR works so well. So we really are grateful, and qPCR is a really, really powerful technique that has a lot to deliver.
It's kind of interesting the way we titled the title of this webinar is sort of sets up dPCR versus qPCR. And sometimes I think when a new technology comes out, it can seem like, well, it's supplanting or it's competing. But is there a space for both technologies in the market? It does sound like that might still be the case. Boshen, would you like to start with this one?
I definitely think there's place for both in the market. It's not a either or situation. I would say the digital PCR and qPCR are complementary to each other right now, because qPCR offers greater throughput and more cost-efficient method, whereas digital PCR brings to the table higher precision and absolute quantification. And also, just like with any new technology, there is a period of adaptation, and people need time to become more proficient in using this technology. Just to add to what Jim talked about, digital PCR, because it uses a different analysis method, it will take researchers time to get used to how to analyze the digital PCR results.
I'm going to keep moving on to this next question that I have for you. I'd like us to talk a little bit more about the analysis component of these technologies. And the first question I have in this regard is, what are some of the main applications where dPCR performs qPCR and vice versa? Jim, why don't we start with you for this one?
I guess you've got to, I mean, if you want to dig down a little deeper in a minute, we can do, but it has an improved precision. It's more precise than qPCR, digital PCR that is, and I can explain why if you want there. It has the ability to, if you have mixtures of targets where you have very closely related targets, such as single nucleotide variants, it has an increased sensitivity there. It can measure lower fractional abundances or lower percentages, for example. It has a unique ability to measure linkage, and again we can come to that, because digital PCR is intentionally measuring single or near single molecules in those partitions. Whether you're measuring 10,000 or 5, it's still measuring near single molecules. So it has very much more confidence when measuring small amounts, trace analysis, as long as you can get that into the reaction. And then an area of particular interest we have is the accuracy and ultimately the reproducibility. It's much easier for you and I to get a comparable result with digital PCR than it is with qPCR. But again, you have the qPCR dynamic range, the ability for the qPCR to do high throughput, vast, huge numbers of runs. Some of the main advantages that I would think with qPCR that still digital PCR struggles to or would struggle to compete. I guess the other area potentially is more decentralization of instrumentation. A lot of the qPCR instruments are now being taken out of the lab to provide nearer, again, in diagnostic, in environmental, in food testing, and lots of examples in veterinary testing, where the instruments are being taken out to be closer to where they're needed. And those are capitalizing on qPCR formats. But I will comment, I guess, that potentially digital offers itself as a better method for a nearer scenario, decentralization or point of care, because the output is arguably simpler. We may come to this. It's simpler for an algorithm to predict has it amplified or not when compared to a qPCR amplification plot.
Yeah, I think on the topic of digital PCRs, what basically the application I think most take advantage of the higher resolution, sensitivity, and absolute quantification are the ones where you would prefer digital PCR. For example, a copy number variation assay is where the resolution is very important. That would be a good use case scenario. And the high sensitivity lend itself to rare mutation detection. And also another factor is by partitioning down the reactions, it sort of dilute out the inhibitors that might be present in your samples. So this makes it good for detection of RNA or microbes and viruses, where inhibitor plays a big role that affects qPCR results.
And Jim, I'm going to come to you here because during your last response, you mentioned differences in terms of precision and targeting, detecting rare targets. And so I do want to dig in a little bit more on those points. So how about we start with precision?
So those of the audience who've dealt with qPCR will, if they're trying to conduct a measurement, they need to optimize the assay, and obviously they'll want it to be having a nice linear range. But also the repeat experiments, the coefficient of variation or the range you might get in the CQ values, or if you convert it to copy number, will depend on your optimization and how how repeatable it is, and that will also in qPCR can vary from day to day. It will certainly vary with instrument. Perhaps not so much now, but when I was doing my PhD, I remember you must vary with whatever you had for breakfast. Yeah, so it's very much dependent on the run and the relationship in those different tubes and how they're developing the fluorescence, and that can vary quite a bit. It also will vary on your pipetting, because your pipetting of the sample will also add to repeatability variation. With digital PCR, you still have the pipetting, but when it comes to the actual technique variability, it is actually dictated by the distribution of the molecules across the partitions. And so you can use this to predict the precision. And it is scarily fixed. Yeah, it really does work because as long as the amplification occurs, it doesn't really matter how efficient the PCR is, as long as you have peak fluorescence to distinguish the positive and negatives, and we have advice in the digital MICA guidelines on how to do that. Really is much less susceptible to this PCR efficiency effect. And actually now with that precision and with the huge numbers of partitions, I mean, when we started, we had like 700 partitions 15 years ago, but now we have tens of thousands or more. You really are able to get exquisite precision that is reproducible. The extent that we are now able to measure what we cannot make. What am I talking about? So my group's looking at increasingly small differences, increasingly small fold changes. And we were able to we got below 10% or 1.1 fold. And then we were going lower, and suddenly the results were going all over the place. It's like, oh my goodness, there's something wrong. And we spoke to our colleagues in the organic chemistry group down the corridor who started laughing at us, because basically with digital PCR, we've gone through the tolerances of the pipettes. So we cannot make a 1.05 fold mix. We can only approximate it. The digital PCR is able to measure that. So we have to now switch to gravimetric measurements of the actual different materials in solution, so we can work out what we aim to get and what we actually got. And so that's how powerful the digital PCR is. Now, my final point on this, and hand over to Boshen, is the audience might go, when am I ever going to need a 1.05-fold difference measurement in such a scenario? Well, we already mentioned a copy number variation that may be occurring either in looking at liquid biopsy measurements for perhaps certain cancers, or genomic or fetal aneuploidy in maternal blood. So there are situations where we can think of examples, but there are also areas we may not realize yet because we have never been able to do this. So really, that precision is it changes everything in terms of what we can do with these instruments.
And Boshen, I wanted to bring you in here.
Yes, in the copy number variation assay, yes, the difference between, just a 1.1 fold, 1.05 fold difference, it could impact the result. There's also like other applications, such as minimal residual disease detection, MRD detection, where the ability to pick out rare variants that's present for at 1% or less of allele frequency is tremendously helpful. So I agree with you, Jim. This will unlock a lot of possibility that we have not thought of before because of the limitation.
And the other thing I wanted to flag, Jim, that you mentioned too, was the importance of the accuracy and the reproducibility. And I wondered if you wanted to elaborate a little bit more on that point.
Certainly. And I work at a National Metrology Institute, so this is kind of what we're paid to look at. But I personally think this could be the big thing that digital PCR can provide to the research and applied community. Obviously, the precision and the rare variants, these are all important new things you can do. But actually, this dry reproducibility thing really could be the thing that changes everything. Because at the moment, if you're looking at qPCR, if you and I are going to make a measurement, I'm in the UK, you're across the pond in the United States, we've immediately got to think about how are we going to make that traceable? What are we going to use to link those qPCR measurements? You remember I said it's highly variable. We've seen now in a lot of the work we're doing with collaborators across the world, including collaborators at the National Institute of Standards and Technology, NIST in the US, that we're able to get reproducible results by just sharing protocols. You don't need a traceable calibrator anymore. We're getting very good agreement just by running protocols, and it's quite tight with the SOPs. So this allows us to suddenly we're getting agreement where we've not been able to have it before in absolute measurements. Yeah, so we have relative measurements where you can potentially get agreement because you're comparing one thing against another, but you've lost the magnitude. Well, now we have absolute quantification, potentially with very high accuracy, which even if we combine extraction, we're finding very good agreement. So suddenly we've never been able to do that before. And it's subtle and it might not be obvious, but that means you and I we're going to be measuring the same thing with very, very comparable results. Now, the next question is, well, okay, you're measuring the same, you're making very similar measurements, by which I mean generally within twofold of each other. Twofold, and that is incredible in molecular measurement. It would admittedly send physicists into a spin. We've got work to do to try and bring it down. The next question is accuracy. Are we measuring the nucleic acids that are present? And we're increasingly of the belief that that can be done both with DNA and RNA, where we're using orthogonal methods like isotope dilution mass spectrometry and are able to show we are getting very good agreement with these SI traceable methods. So not only are we getting good agreement, we appear to be measuring in many cases all or nearly all the molecules with a certain confidence, of course, that are present in that tube. Again, these are things we've never been able to do before. So to some degree, what that means, it really is the sky is the limit, I think.
Yeah, absolutely. Absolutely. Boshen, I'm going to come to you for this next question. I want to talk a little bit about data analysis. How does that differ between dPCR and qPCR experiments?
Yeah, and I'll probably need help from Jim a little bit because you have more experience actually running these assay, especially digital PCR assay. So the qPCR, you're looking at the CT value. You're looking at where the amplification per curve change from enter into exponential amplification phase. So it is something that you often rely on the software to determine, algorithm to determine the crossing point. Whereas digital, you are looking at the endpoint fluorescence, essentially trying to sort out between positive and negative partition. And that also, because the fluorescence is not going to behave perfectly, you're not going to have like a completely dark and a super bright partition most of the times. So the fluorescence level is still going to have fall within a range, and that will require either the algorithm for distinguishing between positive and negative partition, or more in a more complex sample, it will require some expertise to distinguish too.
Boshen's point really stands out is when you start to multiplex. And you can multiplex with qPCR and digital PCR. qPCR requires quite a lot of optimization, and may work perfectly. And again, forgive me for going back to COVID-19, but it's the example that everybody knows. A lot of those diagnostic assays were multiplex. They didn't quantify though. They hadn't been optimized to quantify. And so actually optimizing a qPCR to be both multiplexed and to quantify is very challenging. With digital PCR, it's much simpler, because again, you are essentially setting bins a bit like flow cytometry. You're essentially able to look at different colors, and look at the positioning of the partitions and how they move. And certainly Boshen is correct that sometimes you can get grayer areas, and so there's challenges associated with setting the thresholds of the different populations of partitions. And well, I mean, the digital manufacturers are lots of plexing now, of 5, 6, up to 7 plex in some of the options, and so there's challenges there for the analysis. But again, they tend to follow the DNA moves into the partitions generally in a random fashion. And when it doesn't, that can also be used to look at linkage, because if you look at more than one molecule and they move together, they must be linked. And you can do all sorts of clever things with that, look at drop off, and there's so many, there's so many additional advantages you can have with the digital PCR when it comes to things like multiplexing. And the data analysis is, I guess, coming back to Boshen's point, with the qPCR, you're looking at a relative difference, and you have to factor in the efficiency of the PCR. What does a cycle difference of 1 mean? Is it a 2-fold, or is it 1.8, depending on the efficiency? And so that will impact, and you can ignore that and assume it's 2-fold. Perhaps you shouldn't, but you can. And that will impact the magnitude that you're measuring. With digital, you're just counting the positive partitions or the negative partitions, so that you don't have these additional considerations, which is another reason why it's much more reproducible.
Excellent. And you started to touch on, I think, what my next question was going to be, in terms of the differences in the multiplexing capabilities. Was there more you wanted to add to what you've already said in that regard?
I think that now is a time. I mean, I get asked a bit about the multiplexing. We're learning at the moment of some of the challenges associated, and they are, how do you can think about 2 colors or two axes of a plot, and you've got the human brain can easily look at different distributions. And again, if you've not done digital PCR, you can think of a flow cytometry plot, and you can see different clusters moving around, and the human brain is very capable of doing that in two colors. 3 colors, maybe, if you position it a little bit and maneuver it around, you can imagine some of the principal component analysis that occurs. But some of these now we're talking up to 6, 7 colors. Somebody's going to have to help, because the human brain is not able to cope with that. So these are some of the these are going to be the exciting challenges we have as we go forward. And importantly, I mean, the speed with which you can develop these PCR is really quick compared to some other analytical methods. And so I can see these types of strategies potentially being deployed, where you don't need as many markers as a next generation sequencing solution will provide you, and phenomenal technology there, but you do need a number with a certain level of quantification. And so this is an area where you could see, with multiplexing, what we call higher order multiplexing, where you measure more than one analyte with the same color by having probes of different concentrations, even though they've got the same fluorophore. That can increase the plexing capabilities. So there's a lot of, watch this space, I guess, because I think this is going to be an area of interest where these methods are developed to kind of increase the multiplexing capabilities.
Boshen, I wanted to just throw the floor to you if you wanted to add some commentary there as well on the multiplexing question.
Yeah, I think from an instrument perspective, the qPCR and digital PCR multiplexing capability is similar, because digital PCR, the optical detection technology, it's built upon successful and long-tested qPCR instrument technology. So we're seeing, with digital PCR, we can certainly have a capability of reaching, 6 separate channels, which is similar to that. That's, I would say, like a practical practical plexing need for most labs.
Excellent. And Boshen, I'm going to stay with you for this next question. I'd like to talk a little bit about cost, just in terms of how these technologies differ when we're speaking about cost. And I'll let you respond how you want to that question.
Yeah. Just because digital PCR is still a new emerging technology, obviously at the initial stage, the cost is going to be higher than a more mature technology like qPCR. This mostly comes from the fact that qPCR is more has more throughput. You can run up to 384 samples on a single plate. That's at this point, digital PCR instruments don't quite have that capability yet. So the cost on a per reaction basis, if you just look at a run, it would be higher for digital PCR. But I think there's more economical saving coming from the more reliable results, precision that you get. So without the need to to excessive amount of calibration of instrument variability. So those are areas where you can save reaction, which I believe is more comprehensive look into this. And also the hope is that with time, as the technology becomes more mature, more adoption, the cost will be brought down to that of a mature technology like qPCR.
So as we start to sort of bring this conversation to a bit of a close, I wanted to touch a little bit on some of the key innovations that are currently being made, both in terms of dPCR, and I mean qPCR is established, but I'm sure there's still work going on there as well. So some of the key innovations and what the future might look like for both technologies. Jim, you started to touch on that a little bit in one of your responses. So I'll start with you, and then I'll come to you, Boshen.
So if I think of an innovation that might not be so obvious with digital PCR, we haven't quite touched on, that would be, it's linked to the linkage. It's where you're able to measure the presence of something you cannot see. And so here's an example. So with the linkage type strategies, you can measure the pairing of molecules. And so basically you compare two assays and they will go together in the partition because the DNA links them. But what we can do is what's called a drop-off assay. And so basically you measure the binding of a probe when it's binding to one sequence, or the binding of another when it's binding to another sequence. But if there's a situation where you may have a scrambled sequence and you have no probe binding, but because you can see the molecule with another probe, which is in a conserved area, this allows you to look at heterogeneity. And a great example is CRISPR analysis. And so where you're using CRISPR to change something, you might want to insert a sequence. You can measure the insertion, you can measure the non-insertion, but digital also allows you to measure the scramble, where it's tried to insert it and it's called a non-homologous end join. And so you have a kind of new sequence that neither probe can see. So this is a really elegant strategy because you can see, because you can predict where the groups of partitions will be, because you know the random distribution of the molecules are being distributed accordingly. You can predict where the molecules are ending up that do not have either probe. This is a really elegant strategy for actually allowing you to look at things, an accurate measurement of the signal you're not measuring, if that makes sense. So this, I think, is a really, really unique angle that, again, you can't really do with other methods.
And Boshen, I'll come to you for the last word here.
So I think the power of digital PCR comes from the ability to partition reaction down to many small mini reactions. So more partitioning will lead to more confidence in the results. In that front, at Roche, we have the digital light cycler that we launched in 2022. Which offers 3 different nanowell plate configurations that offers partition ranging from 20,000 to 100,000 that's suitable for different applications. Our nanowell technology improved the partitioning and also prevent amplicon contamination that leads to more confidence in your results. And this digital light cycler instrument is also equipped with a 6-channel optical analyzer, which is similar to our qPCR offering. And on the qPCR front, at the end of last year, we just launched the Light Cycler Pro with enhanced temperature uniformity and improved the software algorithm. It has a 7-channel optical capability, and both of these instruments are designed and labeled for research and IVD, and we're very excited to support both the qPCR and digital PCR going forward.
Thank you both. This was a really great discussion. I certainly learned a lot. And I'm sure our audience did as well. So thank you both for such a great conversation. In just a few moments, we're going to move into a live Q&A session. So if you have not done so already, please submit your questions now. You can do so in the Ask a Question panel on your screen. Before we begin the Q&A, I'd also like to let you know that this webinar will be available to view on demand in the next 24 to 48 hours. So please keep an eye on your email, and please do share the link with your colleagues who might not have been able to join us live today. All right, just give us a few moments to transition to the Q&A. See you shortly.